I was unable to do this first part.
Okay. So anyone who would like to help with what they did or what they wrote would like to help Arpit first?
No, sir, I'm clear with what they did, but like what is transformation was my doubt only.
Okay, so first regarding that only, anyone
Yes.
who knows what is transformation? I'm sure someone might be knowing what is transformation. We have the best of two batches here. People, what is transformation? So we learned about Griffith's transforming principle, right?
Yes,
So
sir.
for me also, even I knew the second and third part, but
Okay.
then that first one wasn't direct.
Yes, so
Yeah,
like
so-
transformation has various meanings. So what we had to actually write was very difficult to
But
understand.
that's why we included Griffith's experiment term so that you will know. So if you are confused between the recombinant DNA technologies transformation, we transform bacterial colonies, right? We segregate transformants from non-transformants.
Yeah, but that's very different.
That's exactly same. Okay, both are the same thing. We are just using that whole concept to do biotechnology. Transformation simply means, as the word says, to transform into something which you were not. So if a bacteria, let's talk about biotechnology. So we give bacteria a gene from outside through plasmid. We give bacteria a selectable marker as well from outside through a plasmid. After taking that plasmid, the bacteria becomes something that it was not. Earlier, the bacteria was not producing insulin. It was not resistant to antibiotics. After transforming it with our recombinant DNA, it started producing insulin, which is not a bacterial gene. It's a eukaryotic gene. Second, it also started showing antibiotic resistant to ampicillin, kanamycin, tetracycline, whatever we have given. So the bacteria got transformed into something which it was not. What Griffith showed was the same thing. So these kind of questions are to test your concepts throughout biology. What Griffith showed was that there were two strains of Streptococcus pneumoniae, S strain and R strain. R strain was not lethal. Rough strain was not lethal. S strain or the smooth strain was lethal, correct? And that was because of a particular gene that produces a mucus. Yeah, a mucopolysaccharide layer on the cell wall that makes the bacteria look shiny and smooth. So that is what gave the name S strain. So S strain was lethal. It used to kill by causing pneumonia. R strain was not lethal. It used to stay dormant and not cause pneumonia. So what Griffith showed, so transformation is the process by which any bacterial cell takes up foreign DNA and transforms to show the abilities which it did not have. Write it down if you want to, because now can you relate that in both biotechnology and in Griffith's principle, the same thing is happening? There is a bacteria that is taking up a foreign DNA and getting transformed to show some abilities that it was not showing before.
So can you dictate it once again?
Yes, transformation is the process by which bacterial cells take up a foreign DNA / gene, take up a foreign DNA / gene and show different abilities provided by that gene or DNA, and show different abilities provided by that gene or DNA. As an example, you can write resistant to antibiotics, upon transformation through plasmids. Resistance to antibiotics upon transformation through plasmids. And second example you can write, the R strain of Streptococcus pneumoniae transforming into S strain. The R strain of Streptococcus pneumoniae transforming into S strain upon taking up DNA fragments from the S strain. Upon taking up DNA fragments from the S strain. Is it clear, everyone? The concept is clear?
So this was related to biotechnology.
It is related, it's related to naturally also bacteria transform. No, Griffith showed that naturally without humans doing anything, bacteria can transform from one type to another. We do the same thing in biotechnology using plasmids, where we are forcefully transforming them into something else for our benefit, right?
Yes.
So that's what the whole thing is. Throughout the year, I have told you to connect knowledge. It's not like whatever you read in biotechnology stays in biotechnology from page XYZ to page XYZ. It's biology, it's science, right?
Yes.
Okay. Anyone else who has any other doubt?
Sir, the 12th question.
12th question. Yes. What is meant by ozone shield? So ozone, you know, right, people? So
Yes, sir.
chapter 16 is not included in your board, but as I said that some important concepts from even the deleted chapters we have tested in this test. It has no harm because, you know, even if you don't attempt it in my test, I'm not going to judge you on that point. But if you do, it's good. So these are very basic day-to-day general science questions. So
Yes.
ozone shield is, so you know that in the in our stratosphere, in the atmosphere, we have ozone, right? Towards, like, which makes a shield around the earth. Ozone is O3. Okay, 3 molecules of oxygen bound together. So there is ozone, and ozone shield protects the earth from UV radiation. So it does not allow UV radiation to reach the surface of the planet, because UV radiation, you know, sun radiates in all wavelengths. Sun is basically a source of electromagnetic radiation. It does not only radiates light, which we can see, it also radiates other wavelengths which we cannot see, X-rays, gamma rays, ultraviolet rays, etc., etc. So the ultraviolet rays are so small in their amplitude, their wavelengths are so small that they have enormous energy. Okay, so ultraviolet is even smaller than the violet range of light that we can see, and it has the most energy. And that energy is enough to penetrate the cells and cause harm to the DNA in our skin cells, okay, in our retinal cells, and can cause skin cancer or sunburns. So from that, ozone protects us. How does it protect us? Ozone absorbs this UV because it can absorb that energy and prevent it from going to the surface. And name 2 ozone depleting substances. One is CFCs, chlorofluorocarbons. When we did not know its effects on ozone, chlorofluorocarbon was very, very readily used. Even when I was in school, it was used in air conditioners, car ACs, and refrigerators. And it started depleting ozone very, very fast. By the time we realized, scientists realized that CFC is not a good gas to use as a coolant, the damage was done. So they have to ban CFC use. And now ozone is healing again. The good news is that ozone is healing. It has not healed completely, but still it's better than what it was 20 years ago. So there was a hole in ozone, which was at the north and the south pole, very big. And how do ozone depleting substances affect ozone shields? They basically react with O3 because O3 is a reactive molecule. It's not very stable. That's why it's not present in the lower atmosphere. It's present in the upper atmosphere. O2 is stable in the lower atmosphere that we breathe in. And remember, if we connect knowledge here, everyone, remember I told you that oxygen is not a very good thing if we think about it. It's true that we cannot survive without oxygen. But if you go back to evolution of life, remember I told you that oxygen, when it started coming in the atmosphere through algae, that was called great oxidation event. And when plants started rising up, the chloro, you remember the chloro ancestors of plants from which bryophytes directly came, and then came others. So they started pumping a lot of oxygen in the atmosphere, and due to which oxygen came as a stress, a toxic stress to the cells. Cells have to adapt to oxygen. Even today, if you give more than required oxygen to any of our living cell, that cell will die. It's not like the more oxygen you give, the better it is. We can only sustain within a narrow range of oxygen concentration. 100% oxygen will just oxidize everything in the cell and the cell will die. So oxygen is a very reactive, todu-podu molecule. It can just break down things. So what these CFCs do, they go and interact with O3.
Then when, like, for example, you're stressed or something, then people say take a deep breath so that oxygen gets into your body.
Yes, and you start forming energy. So howsoever deep breath you will take, you will not inhale more than 21% oxygen, because that is what is present in the atmosphere, and that is important. We have evolved and adapted to that oxygen. I am saying that if I give you 30, 32, 40% oxygen, it is not good for you. It will be a stress, a toxic. Oxygen toxicity is a concept. It will be toxic to your cells, and your cells will start dying instead of producing energy. You understand, Fatima?
Yes.
Yeah. So what these ozone depleting substances do is that they go and react with O3 and break it down into O2 plus nascent oxygen, which is O. And if there is no ozone left, then UV can just simply reach the surface and start affecting the life. Okay. Fourth part, right? Yeah.
UV radiations are still there.
Some components of UV radiations are still there, but that is not in the harmful range, which will start killing the life, because all life survives everywhere, even on equator, where we have the most sunlight coming, right?
Yes, sir.
Yeah. But without oxygen, it will be direct, high energy, very intense UV radiation that will start killing the life. Fourth part is write one damaging effect of ozone on humans and plants respectively. I think the and symbol is missed. So you can say that on plants, what happens is plant cells start dying, and shoot tumors starts arising. Plant growth stops under UV. And on humans, it causes skin inflammation of the eyes, and even skin cancer if given in very high intensity.
So they can also decrease the yield of the crop.
Exactly. They can, as I said, the growth will stop. So if the growth of plant will stop, the yield of all the crop plants will also go down. Good point. Is that clear, Fatima?
Yes, sir.
Yes. Anyone else has any other doubts or questions from this paper? No? Okay, so I'll stop sharing this and I will start sharing the reproductive chapter. Okay, now bring on your doubts from human reproduction. Who has any doubts from human reproduction? Anyone who has any doubt from human reproduction?
So can you just give a brief
Yes,
about spermatogenesis and oogenesis? I mean, I know
spermatogenesis.
it,
Okay,
but then just like a brief type,
okay, perfect.
like revision.
So we'll go to spermatogenesis and oogenesis. And so you know that first, before I go there, let me just quickly tell you, so ask you some very, very curious and interesting questions. So we know that male reproductive system starts producing gametes when the male body reaches puberty, correct?
Yes.
Everyone agrees with that?
Yes, sir.
Yeah. So what prevents the male body from producing gametes from the birth till it reaches puberty? It has gonads, it has testes, it has everything in place. Why is it not producing gametes, and
So
why?
when
Yeah.
puberty, the hormones increase.
What hormones increase?
Sir, the
Yes.
hormonal level is very low.
The gonadotrophin.
Gonadotropins. From where?
Hypothalamus.
Hypothalamus. So hypothalamus is the region of the brain that is responsible for telling pituitary when to start increasing the secretion concentration of gonadotropins, which are FSH and LH. And another interesting thing is these hormones are common in males and females. FSH and LH is same in males and females, but it works very differently. In males, it works, the target organ is testes. In females, it is ovaries, right? This also tells a lot about the common origin of sex as into male and female body. Okay. Both these two hormones are actually named after females, female functions. Follicle stimulating hormone. Males do not have any follicles because they do not have ovaries. Females have primary follicle that converts into secondary follicle and oocyte, right?
So
Males also do not luteinize, okay, or secrete any gamete. They just release, okay. So luteinizing hormone also is named after how it makes the ovum, only one single ovum, released from one ovary and reach into the fallopian tube. So FSH and LH, FSH goes to the testes and starts spermatogenesis. So till FSH level is low, spermatogenesis cannot begin. Okay, and this is why till before it reaches puberty, males cannot produce gametes. And second thing, LH, it goes and tells the testes to start. Yes, Fatima.
who tells the hypothalamus to increase the production of gonadotropin releasing hormone?
Good question. And the straightforward answer is we don't know yet. There are some ideas that I have in my very limited capacity read about. So somehow, you know, our brain knows how old are we. The age of a human body has markers. So senescence is a concept. So people are studying aging. Okay. The whole idea is these very, very cool but idiotic billionaires want to stop aging, you know, want to somehow create a fountain of youth and want to die young. They have money and they often want to recruit scientists to say, okay, find the reason for aging so that we can reverse aging. Scientists are very happy. It's like, yes, we will find it. Give us money. So they give money, we do science. The idea is, see, aging or senescence is not that a single thing. If you, it's like we have realized by now that it's not like we find one gene that helps in aging and we stop that gene from working. that will stop aging, because that gene might be doing 10 other things in the body. If we stop aging, 10 other things can go wrong. Do you understand?
Yes, sir.
One of that is the regulation in the body. Certain things in our body are regulated to get activated and deactivated at certain age, like in females. So if a female body stops aging at 25 years old, let's say, so will it keep menstruating forever? Because female body should stop menstrual cycle when it reaches menopause, around 50 years of age, right?
Yeah.
So, but this is what you, me as science students can understand. But those who are just mad about stopping aging, want to be immortal, can't understand and think. But the
It's
point is, sorry, what is it? It's
impossible to stop aging. It's like a life process.
it's possible to stop aging, but it's impossible to understand what all will be the implications if you stop aging. It is also possible to reverse aging. People have reversed aging in mice. So mice, when they go old,
Reverse aging means like after
make
you
them
become old, you become young.
again. Yes.
How
How
is
does
that
it
possible?
sound? Yeah, how does that sound first? Sounds cool, right?
That's
How's that?
funny. I
That's
don't
funny.
believe
Yes.
that's possible.
Yeah. So one of the so how will you say that a body has aged? So let's talk about that. So like in humans, mouse and humans are very similar in their physiology. When humans become old, you will see that their hair follicles start losing out. So we start losing hair, whether male or female, right? Our skin starts getting detached from the dense connective tissue and the bones, right? So our skin starts to form what we call it as wrinkles, right?
Yeah.
Our neck skins, every skin start becoming loose. Our muscles start becoming less and less contractible and lose their strength, right? So these are all physiological signs of aging, correct?
Yes, sir.
There is a disease called progeria, which is like a rapid advancement of aging. It is a fast aging. So a 13-year-old, if you have seen that movie Pa, where it's Amitabh Bachchan's movie, where he has progeria is the disease which happens due to one mutation in the lamin gene that makes the nuclear membrane structure. So the nuclear shape gets distorted and the whole cell ages very, very fast. Okay. So 13-year-old boy will be as old as a 60-year-old man. So teeth will start falling, skin will have wrinkles, all at the age of 13. So they die
So
very
does
young.
it mean that that boy will die faster?
Yeah, all these progeria, like kids suffering from progeria, go and search this Progeria, progeria. The movie Pa is based on it. It's a Bollywood movie, Pa, which is based on it. So progeria disease is a very, very fast onset of aging. Okay, physiological aging. So in mice also, when they age, they start losing hair from certain parts of the body. A very old mice will look like hair are lost. There will be bald patches, and the mice will, the snout will look different. They will find difficulty in moving around and all. So scientists are not going into details how, because that will be another class in itself, but they have reversed that physiological phenomena to an extent where the mouse aged and then again the hair follicles start coming back after some experimental paradigm, knocking out some genes, and the bald patches got filled. There was a regain of the hair, the mouse activity improved, and the muscle strength also improved. So that's reversing aging. But said so, we still do not know what it means in terms of the whole cell process. Yes, it's possible to reverse aging. It's possible to not allow any signs of and symptoms of aging coming in. But what could be the other effects, we're not very sure of.
So isn't that just physical, like reversing?
So I'm talking about
What about
physiological
like
reversing only. Yeah.
when like when you grow old, then your metabolism decreases. Can you even reverse that?
To some extent, because if your metabolism has not reversed, see here also, the hair follicle takes a lot of, so hair on your skin, on your head, the follicles are live, right? And they are always constantly growing. Yes or no? Hair always keeps growing. So keratin protein is always put. So all that comes from your diet. So it's all related to metabolism. So it's a clear evidence that if you can reverse this thing and your hair follicles can start again becoming functional, it means your metabolism has again regained strength, right? If your muscles have regained strength, if your activity has increased, clearly means that your metabolism is doing better. You cannot do better with activity, growth of hair and everything without changing your metabolism, can you?
Yeah.
Right? So if our metabolism is bad, we will not be able to assimilate protein and we will not be able to grow hair follicles and grow hair back. Because hair is protein only, keratin protein. Yes, so it can be done, but the point is, so the concept was that our brain knows our age. Okay, a lot of our organs have their own clock, senescence clock. So exactly they know how old are they. And the question that how does hypothalamus know that it's the age and let's start stimulating follicle, stimulating hormone? We don't clearly know everything in molecular detail, but we have some ideas that brain knows the age and this is how it is set. That that answer holds true for every other process, like when your other organs start maturing, when your insulin production starts going up and down, when you are more susceptible to heart diseases, when you are going to stop like menstrual cycle and go into menopause, etc., etc. So body has its own clock. Okay. So these two hormones work on gonads and LH starts the secretion of testosterone, and testosterone is an androgen, which is a male sexual hormone, the most important hormone in the male body that makes the male body male. Okay, it leads to secondary sexual characters, and then testosterone goes back and, like in a feedback loop, the more testosterone is there, the more spermatogenesis will be happening. Okay, so FSH starts spermatogenesis, but it is maintained by testosterone. And one of the most important feature is to make gametes, spermatogenesis, right? This is what you wanted to discuss, and this is also what is important, and I also wanted to discuss in today's class. So anyone?
We did this already.
We have done this?
Yes, sir, in the last class, sir.
Okay, Fatima, you were there in the last class and this was done. Okay, Arpit, yes, Fatima, you were there? No. So Arpit, would you like to give a quick review to Fatima? It will be a good revision for you. And
yes, sir. Sir, from the like start or from
just the figure, yes, from primordial germ cell, what's happening and where it is happening and so on. Anything if you will miss, I will just pitch in and complete.
Okay, so sir, this the images of the cross section of the seminiferous tubule, and so seminiferous tubule is the site of the spermatogenesis. Sir, in the seminiferous tubule, sir, there are Sertoli cells present. There is a lumen, which is the central space. There is spermatogonium. So this spermatogonium, which is diploid in nature, so this undergoes mitosis and converted into primary spermatocytes, which are 2N. So then it undergoes meiosis 1. It leads to the formation of secondary spermatocytes,
yes,
which are haploids. And
yes,
it's the first phase in which the haploids, these cells are formed. So then after this meiosis 2, so this leads to the formation of spermatids. Now, so this spermatids are like haploid round in shape. So these have to be converted into the streamlined proper shape of sperm. So this undergoes a process which is known as spermiogenesis and gets converted into a proper sperm, and the removal of the sperm from the cytoplasm of the Sertoli cells into the lumen of the seminiferous tubule is known as the spermiation. And yes, sir, this is what is left.
Yeah, so one thing that you left is about Sertoli cells. Sertoli cells are the most,
sorry, sir, you can do it.
So you mentioned Sertoli cells in the beginning itself, but then you kind of forgot about them. So Sertoli cells are called nerve cells, which right from the beginning, which is called the spermatogonium cells. which are marked here, if you can see my cursor, these are spermatogonium, and all these yellow matrix, these cells you can see are Sertoli cells. Sertoli cells, because these spermatogonium are not motile, but you can see that they are, while they are getting mature, they are moving towards the lumen, that empty space through which they can travel all the way out of testes. So this movement is facilitated by Sertoli cells, and throughout this time, Sertoli cells give nutrition to spermatogonium. When it divides, become primary spermatocyte, then secondary spermatocyte, then spermatids, and finally when they become mature sperm, the process of spermiation means that they are released from these Sertoli cells and go into the lumen. Okay, and then they from the testes they go into epididymis through retrotestes, which is the network like thing here. Through retrotestes and vasa efferentia, they go into epididymis, and epididymis is the region where the functional maturation of sperms takes place. So remember that this question. So okay, this is the neat question, that which is the site for functional maturation of sperms. Okay, seminiferous tubule will be one option, and most of the time students pick that, but that is wrong, because seminiferous tubule is just for structural maturation of sperm. What is the evidence for it? So a sperm in the seminiferous tubule is structurally mature. It has all the structures, but it is not still very, very competent and viable. So the evidence is, if you directly take some sperms from seminiferous tubule and make them compete with other sperms which are ejaculated, they do very poor. They cannot compete, because they have not been through this training camp of epididymis. This is where they are trained for functional motility, their functional capabilities. Okay, is it clear, everyone? Fatima, is it clear to you?
Yes, sir.
Yeah. Okay.
Sir,
Yeah.
the lumen can also be called interstitial space, right?
No. So there's a difference between lumen and interstitial space. Lumen is the empty space in the seminiferous tubule, because it's a tube. So in the tube, you can only call something as a tube if it has a center space. So lumen is a very thin center space. Interstitial spaces is the space between two seminiferous tubules. So in these testes, there will be many seminiferous tubules, thousands and thousands. And in between the two seminiferous tubules, there will be tissues which are called interstitial tissues. And in the interstitial tissue lies the cells called Leydig cells. Okay. So the Leydig cells are present in the interstitial tissue, which is present between two seminiferous tubules, not within the seminiferous tubule. Lumen is not interstitial tissue. Lumen is an empty space. Is it clear, Fatima?
Yes, sir.
And the Leydig cells, which are present in interstitial tissue of the testes, they secrete testosterone when LH comes and give them the message. It acts as a messenger molecule. It's a hormone. Then they secrete testosterone and other androgens, which leads to sexual maturation of the male human. Is that clear, everyone?
Yes, sir.
Perfect. And now I'll quickly go to about the ducts. Please read the glands and the ducts, and let's go to oogenesis. But before I go to oogenesis, my question to all of you is, when does oogenesis begins?
Embryonic stage.
Embryonic stage of the female, right?
Yes.
So if I ask you, the ovum from which you were formed, when was that produced?
When my mother was in her mother's womb.
Perfect. When your mother was not even born, half of your fate was already decided and logged, arrested at prophase 1. So it's very interesting to think like that. Half of my existence was already in my mother's ovaries when my mother was inside her mother's womb. Okay. And then decades later, when my mother was born, She reached puberty. She married my father. Then my father's spermatogenesis made a sperm that managed to fuse with that ovum that gave birth to me. What a chance event, right? What were the chances of me being born? If you just figure that out, taking that there were millions of ovum and millions of sperms, you will realize that every human born is just a lucky draw, isn't it? So we are all in a manner very, very special. It's just like we don't see our life like that, and we sometimes many humans end up doing things that are very counterintuitive to their special birth, special creation. But anyways, so that's right. In a female, the oogenesis starts before birth. So what happens during puberty? Females have already started making gametes. So puberty for them is not about gametogenesis. It is only for males that puberty is about gametogenesis. What is puberty for in females then? What happens during puberty? Anyone?
The ovums get matured.
Yes. So these ovums, they were not ovum when they were formed in our mother's ovaries during embryonic stage. They were just primary oocytes, just like primary spermatocytes. They were primary oocytes. And remember that primary oocytes were 2n or n. Primary spermatocytes were haploid or diploid?
Haploid.
Diploid. Primary spermatocytes were diploid. And you did not pay attention to what Arpit said. Arpit said that secondary spermatocytes are the first kind of cells that are haploid in the whole process. Okay? Is it clear, everyone?
Yes.
Just because the name is spermatocyte doesn't mean it's a sperm. Okay? So, similarly, primary oocyte is arrested at prophase 1, which means the beginning of meiosis itself. So by that time, it is still diploid. So it stays diploid. It's not technically a gamete. When females reach puberty, that arrested phase is lifted off and the maturation begins. Then it goes the first meiotic division. Make one secondary oocyte and one polar body. You understand? And then it makes the ovum and the second polar body. So at puberty, the maturation of female gamete begins and not the synthesis. Okay. And menarche, menopause, all these things you will read. And it's similar for females. So there are primordial germ cell, which is 2n. There is a oogonium, just like there was a, what was it like? In case of males, what was that called? I forgot the term. Spermatogonium. So similarly, there is oogonium here. Where is it? And this oogonium undergoes mitotic division to form primary oocyte, which is 2n haploid, diploid. And at this phase, when it is still diploid, birth happens and everything gets arrested at prophase. Then till puberty, nothing happens. Yes, Arpit.
This we all did right in the previous class.
I think oogenesis we did not do, right? We did spermatogenesis. Now we are doing oogenesis.
Okay.
Okay. Yeah, so the one difference in oogenesis is that there's a phase called atresia which comes, and oogenesis stops completely, gets arrested at birth. Till puberty, nothing happens. After puberty, so from birth to puberty, just these cells are just dying, sitting there and dying. Okay, but there are so many to begin with, around millions of them to begin with. Even if you keep killing them for 12 years, you will still end up having 60 to 80,000 cells per ovary in females when they reach puberty. So at puberty, this meiosis 1 completes and first polar body and secondary oocyte is formed. Again, meiosis 2 completes, second polar body and an ovum is formed. And ovum is the fully mature egg cell. But another thing that you should pay attention to is that when females secrete, they do not secrete ovum from their ovaries. They actually secrete or release secondary oocyte during ovulation. This M2 completion only happens when the sperm comes and makes the first contact with the ovum. That initiates the formation of ovum. So when the oocyte is released, and what comes into the fallopian tube is not a complete ovum. And when sperm enters, then this polar body is already, so it stays with the ovum. So this second division completes when sperm makes its first contact with the secondary oocyte in the fallopian tube. So the question was, what is the site of meiosis 2 division in the oogenesis process in females? So everyone thinks it's ovaries. It's not ovary. What is it? What should be the site of meiosis 2 completion?
Fallopian tube.
Fallopian tube. Okay, perfect. So this is oogenesis. Organs and structures I'm leaving for you to do. If you find any difficulty, you can bring them. Hormonal regulation is same as of males. The only difference is now FSH will go and do follicle stimulation, and LH will helps in secretion of progesterone and estrogen. And progesterone and estrogen are female sexual hormones. They go and they are ovarian hormones, but they go and act at the uterus. And the menstrual cycle starts when the female reaches puberty. Okay. And about the ovarian, so there are two cycles. One is ovarian cycle and the other is uterine cycle. So this is clear. This graph is clear to all of you. A lot of questions comes from just this part. So to everyone, this is clear. All these mountains going up and down. Anyone has any doubt from this part?
No, sir.
No? Okay, perfect. Okay, cool. So let's do one thing. Let's go and do some questions now. Let's see how much previous year's questions. Okay. So the thing is, what I'll do is I'm giving you 2 minutes, people, exact 2 minutes, 2 to 3 minutes while I'm speaking to go through all the notes of reproductive system. Just gaze through it. And then I will start with questions. I will share the screen in front of you containing previous year's questions from NEET exams and other entrances from the chapter. And I will ask someone to read the question. By the time that person is reading the question, after the person finishes reading the question, we will have exactly 1 minute, 60 seconds going strictly by clock. We are trying to simulate the conditions that you will get in entrance and test you under some pressure. And you will write the correct option, just the correct option in the chat just to me. Is it clear, everyone? Up for this game?
Yes, sir.
Saifullah, Fatima, Mamuna.
Yes, sir.
Yes. So only this chapter, right?
Yes, human reproduction. Let's first go with human reproduction. Okay, today. Okay, your time starts now for a 2-minute quick gazing through. And in the meanwhile, I will share the screen. Okay, everyone ready for the rapid fire? Ellen, Arpit, Fatima, Saifullah. Am I audible, everyone?
Yes, sir.
Yes, sir.
Please confirm your presence.
Yes, sir.
Saifullah, Ellen are present, Arpit is present. Fatima, you ready?
Yes.
Okay, so we'll go in alphabetical order, which is, I think, Ellen, Arpit, Fatima, and Saifullah. Okay, which will repeat. So you will read the questions accordingly. I'll say the question name. Then the moment the person stops reading the question, okay, no need to read the options, just read the question. Okay, and your 60 seconds will start. So can you all see the screen that I'm sharing?
Yes, sir.
Yeah, let me make it a little bigger. So, okay, so I'll go with some difficult, conceptually difficult questions, not very factual ones. The factuals, I think you all will be able to do. And there will be mostly more factual questions that you should know, things like that. Okay. Question 16. Ellen.
The function of the secretion of prostate gland is to
Yes. Remember to be correct also, not just the fast. Arpit, typing just one will be fine. I've received Fatima, Arpit, and Alan's answer. All within time. There is still time. Saifullah, just try to be right.
So this is a NEET previous year.
Not previous year, so many years combined, plus NCRT exemplars from other entrances as well. Saifullah, are you there?
Yes, sir.
Yes, perfect. So I've received all four within the time stipulated. And 3 of you have given right answers. 1 of you has not given the right answer. The right answer is option C. Okay. The function of the secretion of prostate gland is to stimulate sperm activity. Remember, prostate gland cannot inhibit sperm activity. So this is outright wrong. Prostate gland is in male body, so it cannot attract sperms also. And none of these would only make sense if C is incorrect. So C, it is very easy one. Okay. So let's go to other questions. And I hope this time all of you will do their best. Oh, I'm so sorry. Maybe. Yes, perfect. Okay. So Arpit, will you please read? There was one more question which I wanted you to read. Yes. Question number 3. It's just
So
about
So the given figure shows a diagrammatic sketch of a portion of a human male reproductive system. Identify the part labeled as A, B, C, and D and select the correct option.
time starts now.
Sir, I sent, so you received.
No. I was waiting for yours only. Time is up.
Oh, sir, I had already sent to, sir. I had sent to host.
Okay, send it to me.
Sorry.
Cool. So again, I have received 4, and out of these, 3 are correct. One of you has made a very, very under pressure silly mistake. It is written that it is a human male reproductive system. You know what they have done? They have just cut this portion so that it looks like similar to female in some manner. I don't know what. So the penis is cut here so that it does not distinctly look different. So it is option A, which is correct. It can't be option C. It talks about A saying ureter. A is not ureter. Okay, ureter is these 2 which are coming from kidneys. Remember that A is coming from below, and only thing which is present below the penis is testes, right? So A is vas deferens and not ureter. Okay, so C and D goes outright rejected. It's A or B. Then you go for the second one, which is same. Third one is the decided one, bulbourethral and prostate. The only thing you should remember is bulbourethral are 2 small at the edge. So the D is bulbourethral, which makes A as the right option. Okay. So this is just a test of pressure kind of a situation. If I would have given you full time, none of you would have chosen a wrong option, but one has under pressure. Okay. Question number 8. Fatima. So remember the sequence: Alan, Arpit, Fatima, Saifullah. Yes, Fatima, question 8.
If for some reason the vasa efferentia in the human reproductive system gets blocked, the gametes will not be transported from.
Time starts now. Don't fall for the devil, Arpit. No need to be fast. You have 1 minute. Be correct.
Sorry, sir.
Yes. Remember, I won't be sitting besides you in the entrance to tell you this, okay? So keep your nerves under control, remember. Perfect. All 4 of you have given the right answer in very fast. Perfect. The right answer is option A, testes to epididymis. Okay. Instead of vasa efferentia, if they have written vasa differentia or vas deferens, then the right answer would have been
B.
none of the above. Vas deferens comes after epididymis. So it would be from, if vas deferens is blocked, then it would be from epididymis to urethra. Okay. Because in between is vas deferens, or epididymis to sperm duct. But none of these option lies here. Okay. That's what I was testing. Okay, Saifullah, you ready? Question number 12.
Given below are the 3 statements, each with 2 blanks. Select the option which correctly fills up the blank in any 2 statements.
Yes, perfect. Time starts now.
Sir, in the B option, sir, the answer for like dash for A is not given.
Yes, right. So that is a trick, no? In A, only for A and B statements are given. In B, only for B and C statements are given. In C, only for A and C are given. And in D, only for A and B. That's what you have to decide, which one is correct. So time is already up, but since it is these kind of questions, you save time for, where you do the other one faster. So I'm giving all of you 30 seconds extra. Okay, so the time is up and I have received all the 4 answers just in time. 2 of you have selected the correct answer, which is option B. B is correct, not C. Okay. Why C is not correct? Let's see. So B
Because
is correct.
Scrotum. That's wrong.
Sorry. Yes, Fatima.
Last it's written scrotum, which is wrong.
That's wrong. Yes. So in C, everything is right. The scrotum is wrong. Sorry, even the urethral meatus is right, wrong. So this is right. In C, option A is correct, but B, both are correct, because statement B is the seminiferous tubules open into the vasa efferentia through retetestes. Okay. Option C is the enlarged end of penis, called the glans penis, or simply the glans, is covered by a loose fold of skin called the foreskin. And this foreskin can be surgically removed under some conditions, disease conditions, and also culturally in many parts of the world. That's called circumcision, right?
So,
Yes.
so like what would be the dash, complete dash for A? So one would be, I guess.
Yes,
So
each
totally.
seminiferous tubule is lined on its inside by two types of cells. This was exactly what we were discussing just now. Inside of seminiferous tubule, there are only two kinds of cells, spermatogonium and certoli. Outside
Yes.
the seminiferous tubule is Leydig cells. Okay. So
Yeah.
I think for A, the correct answer would have been this, spermatogonia and certoli cells. I think on the basis of which many will choose C as the right option, but that's not the right option. Do you understand, people? So pressure can make you do mistakes unnecessarily. This is what this training, these classes are for. So we'll make all the mistakes here, okay? Okay, so coming back to the sequence, whoever is next should read 26. A little bit of imaginary question, but yes.
A sectional view of mammary gland shows.
Yes, time starts now. This kind of question has come actually. Okay, perfect. I've received all the 4 answers and in time, and all the 4 answers are correct. The only trick here is that antibodies can't be seen in any view. Sectional, sagittal, whatever you make, antibodies are molecules. You cannot see molecules. So rest everything makes sense. So option A is correct, 1, 2, and 4. 3 is wrong. Okay. Is it clear, everyone?
Yes, sir.
Yeah. Now let's come to some important conceptual questions from spermatogenesis.
So,
Yes, Arpit.
so can you tell the answer of question number 27, sir?
27,
yeah.
yes. Yeah. So this is simple. Milk secreted from the cells of alveoli of mammary lobes reaches
Oh,
nipple.
okay, okay. It is easy. I thought it's something like gene
Right.
is given.
No. It just name codes it L for lactiferous duct, M for mammary duct, T for mammary tubule, and A for ampulla. So you have to
Yes,
find the
yes.
following order. Simple, right?
Yes, yes.
Since because we have discussed it, can you tell me which is the right order? It always starts with tubule, duct. So remember that TM, like mammary tubule, mammary duct, mammary ampulla, and finally it will reach the, what is?
Lactive.
Yes, right. The
Selective.
lactiferous duct,
Yes.
L. So it will be option A, TMAL. Anyways, so question number 29. Who was reading? Arpit. No, Alan.
2 and no, sir, it's not.
It's you?
Yes, it's my time.
Okay, cool. Read. 29.
2n equals to 16 is in a primary spermatocyte, which is in a metaphase of first meiotic division. What shall be the total number of the chromatids in each of the secondary spermatocyte?
This is a beautiful trick question. I must say.